Namaste. Hamesha Krishna Vidya. I Jibin Preeti Singh, Faculty of Pharmaceutical Science, JVWU-SHORT University. Today, I will discuss about the topic or some short introduction of the microparticles, microcapsule, and microsphere. So, the novel drug delivery system. This topic belongs to the novel drug delivery system theory for the B.Pharm VII semester. So now we come to the what is the introduction definition. A solid colloidal particle composed of natural, synthetic, or semi-synthetic polymers. Size ranges between 1 microgram to 1000 microgram. They can be used as vehicle for the delivery of drugs. Uh, drugs can be entrapped, encapsulated, or sometimes adsorbed on the surface. Also, both biodegradable and non-biodegradable polymers are used. Biodegradable mainly used for parenteral administration. So the method of preparation, ideal characters of a method, ability to incorporate higher concentration of drug and should produce a stable product. Product should have a clinically acceptable shelf life and should produce controllable particle size, have good dispersibility in aqueous vehicle for injection. Release the drug in a controlled manner for prolonged period, biocompatible and biodegradable reproducibility. So the polymer is the natural polymer is protein and polysaccharide. Protein is a gelatin is alginate and albumin is dextran and lectins is chitosan and legumin is agarose and vicillin is palulin. So the advantage is biodegradable and biocompatible. So the disadvantage is batch to batch variation and antigenicity, the conditional biodegradability. Synthetic polymers is pre-polymerized and polymerized in process. So the poly-ecaprolactone and poly-N-butyl cyanoacrylate. So the polylactic acid and polyisobutyl cyanoacrylate. So the polylactide and co-glycolate. All these are synthetic polymer. So the preparation of microparticles from natural polymers is first step is polymer solution is dispersed in a continuous medium, vegetable, oil and organic solvent. So the dispersion is achieved by mechanical stirring, ultrasonication and high speed homogenization. Second step, rigidization of polymer droplets and heat denaturation protein and chemical cross-linking. So the third step is separation, purification and drying. The method of preparation, first one is solvent remove technique and single emulsion technique and double emulsion technique. So the conservation and phase separation technique is by temperature change and by incompatible polymer addition and by a non-solvent addition, by solvent addition and by polymer-polymer interaction, by solvent evaporation. So the cross-linking technique is the chemical cross-linking and thermal cross-linking. So the polymerization technique is normal polymerization and vinyl polymerization and interfacial polymerization. So the spray drying and spray congealing, the freeze drying technique and precipitation technique, so the multi-orifice centrifuge and pan coating and air suspension coating and melt dispersion technique. So the route of administration depends on the drug properties and type of disease and disease state being treated age. So the patient conditions, oral delivery, simplest way of drug administration and not suitable if they release drug for the number of days and GI transit time is varies from the 12 to 30 hours, particle less than 10 micrometer are taken up to by pair patches. This may increase the retention time in stomach. So the mucoadhesive polymers are also prolonged resistance time in stomach. There are the parental delivery prepared with the aim of using for parental administration completely released from the drug. Should be sterile and easily dispersible in aqueous media. Fate of microsphere in the body, biological fate is studied by the radio label technique and C14 and I31 and I25 and 99TC. So polylactic acid is microparticles labeled with 14C were administered by parental route. So the most of radioactivity was found in liver, spleen, bone marrow and lymph nodes and lungs. So this is the complete session. This session is powered by the digital version 2.0 Jyoti Vidyapeeth Women's University. And I hope you are satisfied with my digital session. If you have any query, please mention in comment box. I will resolve it your problem by the problem resolving class. Thank you and thank you for the listening.